Thanks everyone for coming to an aspirational neuroscience journal club. It's been a while since we've had one of these, but we're looking forward to having many more this year, either at a monthly or perhaps even fortnightly basis, depending on how we go and how many speakers we get and how keen people are. Um, so today we're going to be looking at this new paper from 2024, Dynamic and Selective Engrams Emerge with Memory Consolidation. But before we start that, I'll just remind everyone why we're here. Um, so the aspirational neuroscience community is dedicated to sort of understanding and achieving this memory decoding challenge, which is the first demonstration of the decoding of a non-trivial memory or some other learned function based only on the static map of synaptic connectivity. So being able to take, a static brain, a preserved brain, the histology of a brain, some sort of static brain, and being able to read out some sort of non-trivial memory, whether that's the route that a mouse navigated through its maze, or whether it's the particular song of a zebra finch, or some other sort of interesting memory from just static structure. That's the hope that that will actually be achievable with the imaging techniques, behavioral techniques, all the sort of techniques that neuroscience has developed in the past few years, in combination with improved understanding of engrams and memory and everything else that's been happening. But before anyone achieves that particular grand goal with its associated $100,000 prize, the community is also handing out annually a set of annual research awards, which are just honoring and highlighting research into how learning and memory are physically encoded in the brain. So essentially milestones on the path to the core goal of memory decoding from a preserved static brain. And there's been a few of those awards handed out in the past based on papers that were particular important milestones. And there's some recordings of those previous journal clubs and information about those previous papers if you would like to see those, if you haven't come before. And my understanding is that there will be another set of awards handed out later this year at a satellite event associated with SFN. And the purpose of these journal clubs is essentially to assess candidate papers for these annual research awards, to look at them and to see if they're, what the findings from them are, and to see if they are, each of the individual papers that we discuss is a milestone worthy of winning one of these research awards. So that is the general idea. Just for those who've joined recently, we're going to do this as sort of a more interactive journal club. So if there's anything you don't understand or any comments you'd like to make as we go through the paper, then feel free to just speak up as we go. Okay, so a little bit of background quickly before we get into the paper. So engrams is the term given to the physical substrate underlying memories that was posited since the late 19th century, that there would be some sort of material that underlied specific memories that was constituted by specific ensembles of neurons and their particular synaptic connections. But more recently, it's been demonstrated that these things definitively exist by things like forced reactivation of particular hippocampal neural ensembles, leading to memory recall behavior, as you can see on the right here from a famous initial study. And also things like erasure of specific sets of synapses, leading to erasure of specific memories. What's unclear, though, is to what degree are the initial populations that are used during memory formation the same populations that are used for memory recall at later time points? Is it just that, like, during initial memory formation, there's some particular set of hippocampal neurons that become the static substrate for that memory going forwards? Or is there some change out in that ensemble over time? Do some neurons drop in or some neurons drop out? So the evidence in favor of there being some static set of neurons in the hippocampus that encode that memory ongoingly is something like the fact that if you reactivate those initial neurons that were formed during initial memory formation, days after the initial memory formation, then you can still see behavioral recall, like an animal that was had the neurons in its hippocampus that were active during a fear experience. If you reactivate those again days later, then the animal will freeze, even in a neutral context. But the evidence against it being just those neurons that permanently record that particular memory is that if you look at neurons that are naturally active during recall at later time points, you see low overlap between the ensembles that were activated during initial learning and those that are active during later behavioral recall. Something like only 10 to 40% of those neurons that were initially active are active later. And this implies that there might be some more dynamicity to the engram over time and the neurons that make it up. So that's the background before we get to the paper. Randall?
Yeah. Oh, you can see my hand. That's good because I wanted to see whether you could actually see my hand up. Um, so in addition to that evidence about the specific ensemble and the overlap between those ensembles, um, isn't there also the issue that prior results have shown that long-term memory, long-term episodic memory becomes progressively less dependent on the hippocampus overall, that you can still have that recall, over years without the hippocampus being necessary for that. So in a sense, there's a question here when we're talking about engrams and we're looking at the hippocampus as the area of study, is are we really looking at long-term memory, or are we looking at somewhat more intermediate term storage, something that's happening between the moment of experience and where the final destination is of those engrams?
Yeah. I mean, it depends. It's important to check how neuroscientists are defining long-term memory for their particular study, because sometimes it means, like 12 hours, 24 hours. Sometimes it means days. Sometimes it means weeks and years. But there's definitely evidence that you see engram formation in the dentate gyrus, CA1, within the hippocampus, and also in the cortex, basically simultaneously during memory formation. And then what happens over time is that the hippocampal engram becomes insensitive to natural cues as days go on, whereas the cortical engrams become sensitive to natural cues as days goes on. So that's a different aspect as well. That's not really what they focus on in this particular study. They're just looking at long-term memory in the sense of many hours to days, like 1 day or so. But yeah, that's also an important context. Aurelia,
Hey,
and then
uh,
John.
yeah, so I would second what Randall says, and I feel like it just has to be the case. And I thought it was already like really well established that that memories essentially get copied from one cortical structure to another, and that the hippocampus like can't be where long-term memories are because you can remove it and it doesn't reach the long-term memories that are already been encoded, like as they call remote memories that are longer than a month old, right? And is
Yeah.
that not like super firmly established or whatever? I thought that
No,
was.
that is no, that is firmly established. But the question is, even in like I guess medium long-term memory, where you see like the hippocampus is still useful, can result in memory recall. The question is, even in that context, within the hippocampus, is it the same neurons that are being used consistently for memory recall, or is there like a change, a drift in the neurons that are used for memory recall within the hippocampus? You can imagine doing the same sort of experiments that we're going to talk about here, but also for cortical neurons over time as well. Um, John and then Randall.
Yeah, I just wanted to ask on the low overlap between ensembles, are those hippocampal ensembles or are they including cortical?
Everything that we're discussing today is hippocampal.
Okay, yeah. So that to me fits with what has been said so far, that hippocampus may be kind of an active pointer system to the longer term. And so maybe that makes sense that the those ensembles would be much more labile. Yeah. Okay. Thank you.
Randall, did you have another point before we go on?
Yeah, just a brief one, because I mean, you're absolutely right, of course, that whether or not this is the ultimate long-term memory store or whether it's something more intermediate and exactly what's happening there and from one to the other, it is a type of memory. So my question, I guess, is because it was great that you explained before going into this why we're having the journal clubs and also reiterating what the memory decoding challenge is about. So when it comes to identifying a non-trivial memory based on the static synapses, which I think is kind of the short version of the challenge. So is there, it doesn't matter in that challenge, maybe Ken and John can also jump in on this one, what type of storage that is, whether this is LTP, whether this is a change of synapses. So when we're saying static synapses from a snapshot like that, does it matter at all which type of LTM it is?
Yeah, just Ken, go for it.
Yeah, Ken can speak to that, I'm sure.
Ken, you're muted.
I'm muted. Thank you. Yeah, in particular, this type of hippocampal memory, which Ariel is going to explain very clearly next, I suspect. There is, I realize that it's eventually going to go to cortex, but it lasts for at least maybe a couple of days in the hippocampus in a behaviorally modifiable form because of the optogenetic experiments that he's showing here, and there's even a consolidation in the hippocampus, which is a key part of this paper. And so I think this is a pretty core memory that should be considered like structurally stable over an intermediate level, and it could definitely be a something that would be a non-trivial decoding of a memory, at least in my estimation.
Cool.
Okay, thanks for clarifying that. I look forward to seeing that.
All right, let's jump into what they did. So this is the paper again. First author, Douglas Tomé, from the labs of Dheeraj Roy and Claudia Clopath. Let's go. Okay, so they start off basically the experiments. The first part's in silico, and the second part is in vivo. So they do some modeling, and then they check whether their modeling results hold up in actual biology. So they start by defining this sort of like simple model of the hippocampus, where they've got some stimulus, basically some sensory neurons that are projecting into the hippocampus, where the hippocampus is defined by a population of excitatory neurons and a population of inhibitory neurons. And the interesting part is the different connections between all of these. So they're modeling each of the neurons as leaky, integrate, and fire simple neurons, but they're giving them different kinds of synapses. So from the stimulus neurons onto the excitatory neurons, they're modeling it as though just a uniform patch of the stimulus is exciting some random excitatory hippocampal neurons. And that'll be relevant because in subsequent experiments, what they're going to do is like have different stimuli that they present to this model of the hippocampus, essentially meaning they're going to activate different patterns of the stimuli neurons and then see what happens to the excitatory and inhibitory neurons. And then they've also got recurrent connections within the hippocampus from the excitatory neurons onto the excitatory neurons and from the inhibitory neurons onto the inhibitory neurons, as well as connections between the inhibitory to excitatory and excitatory to inhibitory. But between these different populations, they've also modeled a number of different types of plasticity. So some of them are long-term plasticity, such as the stimuli onto the excitatory neurons onto themselves recurrently, and the inhibitory neurons onto the excitatory neurons. And some of the kinds are just static, or have different dynamics. And there's like 3 or 4 pages at the back of the paper listing out all the different kinds of dynamic plasticity methods that they tried. Ken?
Yeah, sorry, just really quick. I was unclear reading the paper. Is all of the excitatory and inhibitory neurons that they're showing here supposedly in the dentate gyrus?
They just model this as a like hippocampus in a generic sense. They do list where they took their parameters from. My memory is that they were trying to model it off dentate gyrus as opposed to just CA1 parameters, but I'm not 100% sure I've got that right.
Okay.
I will admit that I found the computational neuroscience, like all the details of how they did the modeling, also confusing. It's a little bit outside my usual wheelhouse.
Yeah, I mean, one of the reasons why I'm asking is because of the later experiments that I think were targeting mainly the dentate gyrus, and I was a little confused because I thought the dentate gyrus was more of not where memories per se would be, where the main synaptic changes would be occurring, more like CA3 or something. But that's
Yeah,
for some
so like no,
more expertise.
yeah, I mean that's right. So like CA3 is where you see like strong degree of recurrence. My understanding is like there's evidence for engrams. Like you can stimulate a particular population either in dentate gyrus or in CA3 or in CA1 and see memory recall behaviorally as a result of any of those. So you can argue about engrams being located in any of these regions or all of them together. Um, but yeah, at least to me naively, maybe Randall knows more. These sort of recurrent connections, I think, are primarily in CA3, but they are generally doing experiments on dentate gyrus
Yeah,
throughout
that's
this paper.
correct. There are recurrent connections in almost all of those areas, but the majority, the vast majority, that's CA3.
Yeah. All right. So that's the initial model. And then what they do is they do a particular sort of training procedure. Um, so essentially, they start off by initially training by activating those stimulus neurons and then observing the activity in the hippocampal neurons, those excitatory neurons. And then they define engram cells as any of the like excitatory or inhibitory neurons in the hippocampus that were active above some threshold firing rate in the last 60 seconds of the training phase. So after stimulus presentation. Those are training engram cells. And we have to keep in mind the difference between training engram cells and consolidation engram cells and recall engram cells because they're always going to define engram cells as the cells that were active in the last 60 seconds of whichever phase they're looking at in response to some particular stimulus pattern. So, sorry, Randall?
This is where you're going to notice how little I actually read the paper. Um, is Are they in all the way through from here through everything else? Are they going to be basing the activity in the hippocampal model and maybe even in what they're recording on rate rather than spatiotemporal patterns of firing?
Yeah, it's always based on rate during the last 60 seconds.
Okay,
With
interesting. All right. Yep.
Yeah, I'll clarify it'll become clear as we go on. It should make sense as we go. Uh, so that was the training. So in this initial first phase, can you guys see my mouse by the way? Move it. Um, Who knows? Okay, cool. So they did the initial training with stimulus presentation, and then what they wanted to do was a consolidation phase. So what they did is for a period of time after the initial training, over simulated hours, they would repeatedly reactivate the stimulus neurons uh, in the same way that they'd been active during initial training. So they represented the training stimulus in a way that mimics reactivation and replay that's naturally observed in like real animals during post-encoding sleep. So if you look at the hippocampus of a mouse, for example, that's done some navigational task, you'll see that the neurons that were active in the mouse brain during the initial task get reactivated during sleep. And that's the idea that they have here during consolidation. Yeah, so they're reactivating that specific stimulus population. And then also during this consolidation phase, they regularly perform what they call probing, which is when at a particular time point, so let's say 1 hour, 2 hours, 12 hours, whatever, they re-show the stimulus again. So again, it's similar to consolidation, where they activate the stimulus population, but then they observe at the end of that presentation which cells in the hippocampus are firing a lot. So which are above threshold, and thus which is the engram that represents that stimulus at that particular point in time as consolidation has been progressing. And I think they do the stimulus presentation with slightly different dynamics to how they're doing it during consolidation, during this probing phase. And then finally, after many hours, simulated hours of consolidation, they also do a recall test, which is similar to the probing, except that they don't necessarily use the entire stimulus. They don't necessarily reactivate the entire set of stimulus neurons that were activated during training or consolidation. They may only do a subset of them or a variety of different new stimuli. So for example, you can see the training one here, the different novel ones, and then there's partial cue presentation variants that you can see on the right. And then in the same way, as they did in probing, they identify the engram cells that are active at that particular time point. That's the core of the simulation that they did as part of this paper. Any questions before we move on to the results? No, good. All right, let's continue. Okay, so this is probably the critical thing, like the critical figure in the paper. What they wanted to know is if you look at the population of engram cells identified at any particular time point by probing or by after initial training, they wanted to see, well, what is the overlap between cells that were active, identified as engram cells at the end of the initial training versus cells that are active after stimulus presentation, so during probing, at any particular subsequent time point, and to what degree do these different populations overlap? And so what they initially found, for example, is that you see the overlap between cells that are activated during each probing test does start to stabilize over time, but initially there's quite a bit of changeover in the cells that are active during probing. So that's what you can see in the solid line. And the dotted line is showing which cells are identified as engram cells the entire way through all of the consolidation phases and probing phases. And it's only a small set of the cells that are initially identified as engram cells that stay as engram cells throughout the entire experiment. Randall, I might get you to hold your question for one moment until I just explain the next few things. So then to make that clearer, what they show is they look at which cells overlap between the probing populations and the training population as a function of the percentage of cells that were active during training. So this is basically asking of each cell, like of the engram cells that we see at any particular subsequent time point as identified by probing, what percentage of those were also identified as engram cells during initial training? And you can see it like starts off, obviously, by definition, as 100% and dramatically drops down over the consolidation phase. Those are cells that they're identifying as drop out. So they were initially part of the engram, but then they dropped out of the engram over time. What you can also look at, though, is cells that were active at a particular probing time point. And you can see which of those overlap between the probing population and the training population as a percentage of cells active at that probing point. So if you're at time 12, for example, and you're looking at 100% of the cells that are identified as engram cells at time 12, then to what degree are the cells that were active during initial training a proportion of those cells that are now active at time 12? And that also declines over time, which basically is implying that there's new cells that are being added in over time. And those they call drop-in cells as part of the consolidation. All right, Randall, I'll get to you now.
Yeah,
after those
so I might be pressing you on something that's too detailed, but I was trying to understand, just because it's a model, I'm trying to understand what is causing different cells to activate in response to the stimulus initially, when we're not talking about a previously already established set of excitatory weights, although of course there will be something there because they've initialized to something. So I'm going to ask about a few things that are contributing. I assume that they add noise to their simulation.
Yep.
I assume that they maybe have spontaneous firing. Not sure if they do. Could be in there. Do you know if they do?
I would.
I mean, if they have enough noise, they will have. So it's almost the same question.
Yeah, they definitely add noise. I'm not sure about spontaneous firing.
And do you know what they did to their recurrent excitatory connections to begin with? What was the initial state of those connections?
They are initialized. Yeah, it's like a random uniform distribution. I can't remember the parameters for like what proportion of neurons they are connected to and what the strengths are for the initialization.
Okay, yeah.
Which is
all right.
Yeah.
And then
sorry.
I guess my final question is, so maybe you already mentioned this in the beginning and I missed it. What was their initial, like, what was the reason for going into this and doing it? Because I'm wondering, so far I haven't seen anything that surprises me. So I'm wondering what they were actually looking for.
I think they're particularly keen on identifying the like dropout and drop in, like the bits that I was just explaining, where they want to see to what degree like the engram representation changes over the period of consolidation. And can they identify how it's changing and like which elements are influencing?
So they're trying to identify the mechanism that causes that. So for example, that in the beginning you might have more different patterns of activity because of what's randomly firing, and then later on, because you have more excitatory weights being established, you get a more stable group in a sense. So are they trying to confirm that, or is there something that they find would be like a surprising finding?
I think when they look during the introduction, they're surprised by the degree of like reduction in engram population over time compared to the initial engram, and they're trying to identify like what are the mechanisms that cause that.
Okay, so that's then involving
and it caused the drift.
Okay, got it. Yeah, thanks.
Okay. So we talked about drop out, we talked about drop in, and this last figure is just showing the intersection between probing neurons and training neurons as a percentage of all neurons over time. And you can see that like the number of cells in the hippocampus that are active as engram neurons just also declines over the course of consolidation. So that's during probing and consolidation, but then they also look at recall where they use partial cues and they see what's happening during that time. So again, partial cues are either partial cues of the initial training stimulus or of the novel stimuli. And basically why they're using these novel stimuli is they want to look not just at like recall, can the animal remember the stimuli it was shown, but they also want to look at selectivity. So will these populations with neurons respond specifically to the initial cue or partial cues of what they were initially trained on, or will they respond to partial cues or other novel cues, so in an unselected fashion? So again, they're observing the engram cells identified by probing at a particular time, and they are measuring firing rates in response to these cues. That's how they're defining engram cells. And so basically what you see is that over consolidation, initially you see neurons firing in response to any of these sorts of cues, whether it's the initial training or the novel cues. But then over consolidation, the neurons become quickly selective for just the training stimulus, whether that's identified based on firing rates or whether that's identified based on the proportion of neurons that are active in response to any of these particular stimuli at a particular time point. So basically that means if you probe at, let's say, time 30 minutes, and you probe by showing any of these stimuli or partial stimuli, you'll see activity that overlaps with the initial, like what neurons were active at the end of training. But over consolidation, they become selective just for the training stimulus. So this is the key of the simulation that they wanted to perform. And then to get a little bit into Randall's question about like, well, what are they actually looking at? How does the network change over time? What they did is they looked at the weights and the changes in the weights between these different populations, both before consolidation, so on the left column at time 0, and after consolidation, so on the right at time 24 hours. And they're looking at all the different populations between non-engram cells and engram cells identified at those time points, and for the different types of connections. And so what they saw, for example, is that if you look at these connections, excitatory connections from the stimuli onto the excitatory neurons, then before consolidation, it's like fairly random, like it's pretty uniform. Although there's a little bit of higher weight between engram cells themselves, which actually I realize I don't fully understand how you can have strengthened engram cells from stimuli to excitatory. Anyway, but the point is that after 24 hours, you see this like much stronger connection between the stimuli and the excitatory neurons, between the stimuli neurons and the hippocampal neurons, as expected. You also see a strengthening of the recurrent neurons in the excitatory connections. But what is probably most interesting is you see this very strong increase in inhibitory activity onto excitatory neurons, both for inhibitory engram neurons onto excitatory engram neurons, and for the inhibitory engram neurons onto non-engram excitatory engram neurons. So the basically inhibition increases substantially across the board. I think that's all the modeling data before we start to get to in vivo. So I'll just check everyone's still on the same page before we go on to the in vivo data. Randall again.
Sorry for asking so many questions.
No, please do.
Yeah. So the state of the interneurons when we look at before, before and after, when we look at the connections from interneurons to themselves, the excitatory connections from interneurons onto themselves.
Hold on, just to be clear.
So the bottom one, right?
Yeah, so that's from inhibitory neurons onto excitatory neurons. Inhibitory neurons onto themselves as an inhibitory onto inhibitory is static. It's the inhibitory onto the excitatory neurons.
Oh, okay, sorry, didn't see that. Yeah. Okay, they're static. So this is all this is from interneurons onto excitatory. Well, no, anyway, it doesn't really change what I was going to say. Sorry. Yeah, it comes down to the same thing. So what we're seeing, the change here, is that it's establishing something in the network that originally was pretty much random, that is typically assumed whenever we start modeling these areas, which is we model it as an interneuronal network that is connected to all of the pyramidal cells and silences them after they fire. That's pretty much what I'm seeing here.
Yes, I think that's the point.
Yeah.
That you're seeing this increase in suppression, and then combining with the what you can see in the top right, which is that you get this strengthening of the stimuli onto excitatory neurons, and I think the idea is you're starting to get more selective over time.
Yeah. It's. I guess I'm reiterating this or pointing it out because I thought that I was looking for a new insight about the role of interneurons, but maybe that's still coming up because this picture is exactly the picture that is always assumed about how things work in the hippocampus and why you get this separated firing of patterns with, the inhibition in between each one of the reactivations. It's because of the population of interneurons suppressing everything right after a pattern fires.
Yeah. I mean, so as not an expert in computationally modeling the hippocampus, what's clear to me that goes on is they want to contrast their modeling results with the in vivo experiments that I'm going to be talking about next. So perhaps they just wanted to have like a clear comparison, even if this isn't particularly novel, but I'm not entirely sure. Ken?
Yeah, I was just going to say that I think they make the case that this inhibitory to excitatory plasticity has to occur for this to happen. So, Randall, if you're saying that there's an existing static inhibitory to excitatory that is setting up this, and their model is not assuming that, but makes it happen. The later experiments are suggesting that that plasticity has to occur while the consolidation is occurring, which I
Okay,
think
so that might be something novel. Yeah. Okay, thank you for clarifying.
All right, thanks, Ken. All right. Oh yeah, and then lastly, they just before I get to the in vivo, they do a few
Recording
consolidation control experiments to sort of validate their modeling. So for example, one of the things they were interested in is we talked about how you need there's evidence that consolidation involves reactivating neurons that were initially active during initial stimulus presentation. That's in actual animals. So what they were questioning was if you block that consolidation by blocking that replay, how does that affect memory development? So what they do is they rerun their experiment, but they block sensory reactivation during the consolidation phase. And what they see as a result of that is the fraction of neurons that are active just stays static instead of reducing over time in comparison to in the initial version. They see that the proportion of neurons that are overlapping during, like as in that are active during probing, stays fairly high and doesn't have this initial change from, like as in doesn't initially change out to a reasonable degree, which proportion of neurons are active during probing. And they also show like a low degree of selectivity during the recall at the end, if there's no consolidation occurring. So the neurons that are activated by the cues overlap to a large degree during recall if consolidation is blocked from happening. They also run a set of other control experiments where, for example, instead of blocking consolidation, they block some of the excitatory plasticity. They show that that also impairs recall. And they also show that blocking the inhibitory plasticity dramatically blocks selectivity. So these being able to only have engram cells active for a particular cue. And there's going to be a lot more on that as we go on. I notice it's 9:40 already, so I might go through these bits a little bit faster so we can have some discussion at the end. I might just skip this bit because it doesn't matter that much. Okay, so how do these results hold up in vivo? Like, do their simulations match what we actually see in biology? So as a reminder of how they do their memory experiments, they do contextual fear conditioning. So that involves taking an animal, initially placing it in some environments, and then subjecting it to some scary experience. Then they wait some period, so an hour, 5 hours, 12 hours, 24 hours. They put it in a neutral context, and they see the degree to which it freezes or moves around. Normally, if you put it after a sufficient period of time into a different context to one in which it was trained in. Whoops, I see that that's mislabeled. That should say neutral context above. Then the animal will continue to groom and move around. But if you put it back into its training context, then it will stop moving and it will freeze if it's had this initial scary experience. And this is like extremely well established, but they just replicate this sort of behavior with their particular experimental setup. And what you can see is that over time, initially, if you just after the training context, so after it's been subjected to these shocks to its feet, if you put it back into the training context, or you put it into a neutral context, they just tend to freeze a lot in response to both conditions. But over a period of hours, particularly after 12 hours, what you start to see is the animals freezing only in response to being placed back into the training context. Whereas if they're put in a neutral context, they don't freeze. So they realize, they can remember that that's not where they had their initial painful experiment. And what they want to do is they want to contrast this actual behavioral results against the selectivity increase that they see during consolidation, where initially during probing, what they see is that any of the cues in their simulation can reactivate the engram population, But over time, you see selectivity for only the training queue being able to do that. So the way in which they actually measured this is they recorded which neurons were active during memory formation, and they did that using this Cal light probe. So it works by if you inject the Cal light proteins, like the required genes for those proteins, into the dentate gyrus, you have a setup where then during contextual fear training, neurons that are active both intrinsically, so that have calcium firing, and that have blue light shone on them, become labeled. So essentially, what you're doing is you are labeling only the neurons that were active intrinsically during fear conditioning and at the time when this blue light was shone on them. And the advantage of that is it gives you like very clean labeling of neurons that are active only at the particular time when you wanted to label. So only during the contextual fear conditioning rather than in their home cage just prior to the fear conditioning or just after the fear conditioning. So using Cal light, they're recording the neurons that are active during the initial memory formation, and then they wait for some delay period. And then they're also going to look at neurons that were active as labeled by CFOS staining. So this is an immediate early gene. This is a gene that's expressed by neurons that have been recently active, and it lets you record which neurons were active just prior to killing the animals, perfusing them, and doing the histology. So basically, they have these two different labels. They have the label for initial training, the Cal light, and they have the label for whatever was happening during recall just prior to killing the animals. So what they're going to do using these two different labeling techniques, one for the neurons labeled during initial training and one's for the neurons labeled during recall, is they're going to contrast that against their simulation results for neurons that were active during training and neurons that were active during probing. And what they show is sort of analogous results between the behavioral experiments and their initial simulation, where if you look at the overlap between neurons that were active during recall and that were active during training as a proportion of neurons that were active during training, what you see is this decline over time in a way that mimics the dropout of neurons that were active during training in their simulation results. You see the same thing if you look at neurons that are active as a proportion of the CFOS, so that neurons that were active during recall. So you see essentially analogous things to drop in, although admittedly it's less clean than with the dropout neurons. And you can also see it as a proportion of all neurons. So this is one of the key things that they wanted to establish, that there was this essentially dropout of neurons that are active during the initial memory formation compared to when they're observing which neurons are active during recall at a later time point, and that there's an analogy between their simulation results and their behavioral results. It's kind of the key thing as part of the paper. I might skip this part. This part is essentially just showing that despite that dropout, if they forcibly reactivate the initial cells, so as in the population that was active during training, that still leads to behavioral recall, saying that despite the dropout happening naturally, the initial training population is still sufficient for memory recall if it's artificially reactivated. And then after establishing that, they wanted to drill down a little bit more. I've got 2 more slides of data, which I'll go through before we can have a discussion. They wanted to drill down a little bit more into what were the actual mechanisms going on in this dropout, drop-in, increase in selectivity process. So they looked at 2 things. They looked at the role of inhibition during recall, and they looked at the role of inhibition during consolidation. So looking at it first during recall, what they did is in their simulation, they blocked inhibitory neuron activity during recall. And basically what they found in their simulation is if they blocked inhibitory neuron activity during recall, then what they saw was like no selectivity of engram cells for the particular cue that they were presented with. So they would respond, but they'd respond equally to the training cue or any of the novel cues. And by analogy, what they also did is they optogenetically inhibited interneurons in the dentate gyrus, the specific population of CCK plus interneurons. And they showed that if they inhibited those during memory recall, that it would block selectivity of freezing to just the training context. So you can see on top, this is showing again the data from before, which you can see that not at 5 hours, but at 12 and 24 hours, the animals only freezed in the condition, like the training context. But if you block CCK plus interneurons, it prevents that selectivity for freezing only in that context. And what was interesting as well is it was specific to that population of CCK plus interneurons. So if you look at some other population of interneurons, such as parvalbumin interneurons, it didn't have any of these same effects at recall time specifically. Might have had effects if they were blocked during consolidation or probing or earlier time points. So that was recall, but they also wanted to know, well, what about during the change out in the engram? So during the consolidation phase, what happens if you block inhibitory plasticity during that time point? So again, in simulation, they block the inhibitory plasticity. And what you see is this sort of just messes with recall rate throughout the entire consolidation time. So it messes with the selectivity and prevents discrimination based on which particular cues are presented. And they also tried to replicate this in vivo. So in this context, they didn't use optogenetics, they used chemogenetics. So basically what they did is they injected the dentate gyrus with with a virus which rendered the CCK plus interneurons sensitive to inhibition by the introduction of a particular non-physiological chemical, C21. So it meant that CCK plus neurons, if they were exposed to the C21 chemical, would now become inhibited. And then they did their fear training, their initial fear conditioning. Then they injected them with this C21 chemical, which is going to block dentate gyrus interneuron, CCK plus interneurons from being active for at least 12 hours after that time point. And what they saw, again, is that this recapitulated the previous lack of selectivity as a result, where, again, on the top is the control, where you can see that selectivity for freezing only in the training context is increasing over 12 hours and 24 hours. But if you block this particular population of interneurons, you see that this selectivity doesn't emerge. Okay, so we've only got a few minutes left, so I'll get into the summary and discussion. So basically what they show over the course of this paper is that neurons are removed from and added to the dentate gyrus engram ensembles as the memories consolidate. So over that 24-hour period. And this is occurring considerably faster than the active to silent handover of behavioral control that happens when you go from hippocampus control of behavior to cortical control of behavior, which takes days, as opposed to this just sort of hours of consolidation. They show that the inhibitory synaptic plasticity is crucial to the memory becoming selective. So in this example, the CCK plus interneurons, but they note that in other brain regions, it might be different populations. So in the lateral amygdala, it might be the parvalbumin neurons that are important, as opposed to CCK plus interneurons. And they say that this is consistent with other findings of representational drift, where neurons that are used for behavioral recall or sensation at a particular time point may change over time. So for example, other examples of representational drift are that specific neural populations that are active in response to some olfactory stimuli or auditory stimuli in the cortex will also change over time. So you present someone with the same stimulus and you won't always see the same set of neurons active. Lastly, and then I'll open this up for general discussion in the last 5 minutes, is the relevance of these findings to memory decoding and the aspirational neuroscience challenges. What this paper sort of suggests is that engrams in preserved tissue, being able to identify them, may require different decoding strategies based on the degree of consolidation that's occurred to that particular memory. So knowing the precise timing, since memory formation might be critical, or knowing what engrams look like at different stages of consolidation might be critical. And it also emphasizes the need to be able to classify, potentially to be able to classify particular inhibitory neuron class. So in this example, it would be important to be able to identify the CCK plus interneurons, more so than the parvalbumin interneurons, which is feasible with immunohistochemistry. I am unclear as to how feasible that is with just looking at morphology. I'll just leave it there. Okay, that's the core of the presentation. Sorry for the rushed overview of the paper. There's quite a bit that I left out as well. But I'm curious as to what people think, whether they had particular takeaways, whether they think this was just actually, showing the same stuff they already expected, as Randall said. Does anyone else have any thoughts on the paper? Thanks for the claps. Randall, go for
it. Sorry, I was hoping someone else would go first just so that I'm not always the one bugging you. So first of all, this does present something new that I hadn't really thought about, because I hadn't thought about exactly why or how strengthening of the connections from interneurons to to pyramidal neurons would make a big difference, as opposed to simply strengthening recurrent connections and having kind of a static set over there. So that's interesting, even though a thing that's missing for me is there isn't really a good explanation as to why this is the method chosen, because you could also create a fairly focused, strong core engram just by sufficiently strengthening the excitatory recurrence on the pyramidal neurons. You could. Now, it so happens that this system has all of these other connections in there, and there's no reason why those shouldn't be plastic as well. And so, yes, they'll be involved. But what precisely? Like, do they have more information about that in their paper? Because there are other things that are also involved when you're talking about interneuronal networks that are super interesting, and where maybe this plays a role, which is all of these issues about, say, having different modes of encoding and retrieval going on, kind of taking turns in every theta cycle in that entire system. And you've got specific types of neurons. You mentioned that it's important to be able to identify the type of neuron. That's probably true, because there are certain types of neuron, for example, that make connections not onto another cell, but onto the terminal hillock, the sort of like of an interneuron that's trying to make, a connection somewhere, and can inhibit the ability to pass an action potential through there, so that you can block a path, in a sense, by activating those interneurons. And that's really useful if you're going to try to run things in different modes, have switches like that. So that's something that's been used in other models, and I won't get into the details here, but I'm kind of thinking that to really understand why this particular method seems to be what the hippocampus is using to make more focused engrams, that may have something to do with the remainder of that mechanism, that stuff that it's embedded in, and not just the little core that's trying to make an engram itself within CA3. Or do they have more in the paper? Because I didn't, obviously
they have a lot. They have a lot more in the paper where they try essentially messing with different kinds of plasticity and seeing how it affects selectivity or memory formation. So they try inhibiting only particular kinds of excitatory plasticity, or only particular kinds of inhibitory plasticity, or messing with the dynamics. And they see, based on which particular kinds they mess with, like increases or declines in selectivity, and like also increases and declines in selectivity at different time stages. So I don't think I can give a super satisfactory answer beyond, it seems like how you tweak the parameters changes the time scale and degree of selectivity that occurs. And I'm not sure in the like actual biological hippocampus how those different parameter trade-offs are set.
Yeah, another thing that was a bit difficult for me in really evaluating what's going on in the paper is that they use the firing rate as their code, because that way you don't get to look at what's happening at a specific phase of rhythmic activity in the region, and phase locked activity, and exactly what's happening when this population activates, then the next one, and then it shuts off that one, and then you get a different pattern for another stimulus, etc. These things are you're kind of at the heart of these hippocampal models. So it was a little frustrating that they're just dealing with firing rate, which is like kind of putting a big blur sponge over things.
I have a vague memory that in one of the extended data figures, they may do a bit of analysis of phase, but I did not look into that in detail. Ken?
Okay.
This is really great. I think it's a bit of a confusing paper, but very, very relevant. One thing relevant to Randall's just last question was their use of this, what was it, CA light, calcium light activation stuff. I'm wondering how narrow of a window that actually gets, because all the previous stuff was CFOS, TTA, or immediate early gene stuff that had just a gigantic time window. And this is a much narrower window that they can tag cells in. I suppose I'm wondering if it's so narrow that you could do phase locking like Randall was just suggesting. Yeah, any idea?
I'm not sure the exact duration, whether it's like seconds or minutes or milliseconds. It was new to me, actually. I'm clearly a little out of date with my imaging techniques because I wasn't aware of Cal Light till I read this paper. So I'd have to go check the papers. But it is a vast improvement over, because I'd heard people complaining about how like previously you'd be using like the initial studies from like 2012 to 2015 or so were using what was it? Something they fed the animals. I forget exactly the compounds that they would use, but basically they would just open up these windows of like an hour or so when they would be labeling cells. Yeah, doxycycline, that's right. Where they'd be labeling everything. So you'd get a lot of like overlap with like, neurons that were active during the home cage and handling and stuff. So being able to use this combination of like, we're only tagging neurons when we shine the light on the hippocampus is like greatly refining down the selectivity of the population identified, which is cool to see.
Yeah, I think the other thing is reference number 9, which I didn't look up, it seemed to be where they actually had developed this model. And so I bet a lot of the details of the motivation of the computational model are coming from that. So let's see, there was one other thing that I'm blanking on. So in any case.
I'll also just flag for those who are new, I allocated an hour for this, so I won't be offended if you want to leave from now, but also feel free to stick around and chat for a bit as well. Sorry, Ken, I interrupted you.
Oh, I remember my last question. So they make the argument that inhibitory plasticity is necessary. Okay, fantastic. Let's assume that is absolutely correct for the moment. Did they show anything about whether it's information carrying? This gets back to Randall's point earlier that if this is a kind of a widespread inhibition, then it may be absolutely crucial. It's a homeostatic mechanism of sorts, but it might not be information carrying like the synapses that are excitatory.
Yeah, I was hoping to see something like what happens if you like knock out particular subsets. So they talk about blocking inhibition or artificially reactivating the neuron. But I would have been curious about like what if you block particular subsets, like whether in a proportional sense, like you block 50% of inhibition or 20% or 50% of the excitatory neurons, or if they could identify particular subsets of the populations that matter or don't matter. They, as far as I could tell in the paper, don't do the analysis required to work out if it's information carrying or not for the inhibitory activity, but I'd be curious to see that. I'm curious as well, like in terms of my own question, which is like this seems like the sort of simulation work you could do to understand like what proportion of the engram neurons you'd need to identify in order to selectively identify a memory, where like they're showing this like over the course of consolidation, you can see that like the memory is reactivated only by the particular training cue as opposed to the partial or novel cues. But like if they start to degrade these engrams or like they destroy a certain proportion of the connections, or if they just can't identify, like they're unable to identify some of the engram cells for whatever reason, I'd be curious to see how the results hold up or don't.
I'd say the next journal club, I think, although it's listed as you, but there's a paper that I'm thinking of that is also kind of engram to engram cell connectivity changes during a learning formation in the lateral amygdala. And I think that one might address exactly what you were talking about. So
yeah, cool. Well, we can, either I or you can do that as we see fit. Happy to mess with the schedule. Randall?
Yeah, you can definitely do experiments like that in simulation to get some idea of, at what point degradation becomes problematic. That would also, that'd be interesting for a lot of reasons. One thing you mentioned at the end of your presentation was that, one of the things we have to think about then is the kinds of memory consolidation that you can pick up after preservation of a brain. I'm not sure if I'm paraphrasing that correctly. Was that what you said, in a sense?
I meant to say that because it's like a demonstration that the engram underlying a particular memory changes, like the neurons in the synapse underlying it change over the process of consolidation. It might be the case.
This is consolidation over a short period of time, right? So
yeah,
it's still kind of like if you lose that, if we just don't include that in, that's not something you would get back. You have a little bit of retrograde amnesia, but not a lot.
Yeah, but the question I guess is like to what degree is this analogous or not in like longer term cortical memory as well? Like do you continue to see this representational drift in cortical memories, or are they like do they eventually become like totally static? Is like something it might be known, but I don't have a definitive answer to that. And when I read a paper like this, I'm start to become a little like, oh, that seems like maybe we should check that. John?
Yeah. Actually, can I just tag on one bit to my question?
Yeah.
So we're still talking about LTP here, right? Because these methods that they used, which were all activation based, not based on, say, actually looking at synapses and seeing whether they've changed, can only pick up on strengthening or weakening of a connection, not whether it's LTP or something else. And LTP can last for days, even weeks sometimes. So this would not be necessarily something you would find if you take a preserved brain and you're looking at morphology, although you might be able to detect it from looking at, if you're tagging proteins. Yeah.
Yeah, that's a good point. John?
Yeah, I guess I'm piggybacking a bit on Randall's last comment. I was researching triplet STDP and how it works, and I read this section in the memory consolidation reshapes engram. I read this sentence, which was really news to me. I'm sure you guys all know it. Transmitter-induced plasticity prevented neurons from becoming silent. So, am I understanding this correctly? That it's these STDP and other non-Hebbian excitatory inhibitory activity that they're really describing as kind of the core to creating the engram, and then the neurotransmitters are like a secondary thing.
I'm not
how do I
sure.
understand that?
I'm not sure how we can determine what we're talking about here because of the, we were looking just at firing rate changes, but STDP, as far as I understand, is Hebbian. I mean,
yes, it is. Yep. They were
yeah,
saying
so,
that
so it's not non-Hebbian.
that's
Which
right.
ones were you pointing to that are non-Hebbian?
Heterosynaptic, which is the post-activation of the postsynaptic neuron, and transmitter-induced, which they're kind of describing as this something that's like a threshold on it creates a threshold level of activity that keeps the whole kind of network active, as I was understanding it. That was page 562 in that first column in page 562 was where I was reading all of this. So my main question is, where do you think that kind of information would be actually encoded in the structure? Or is it all dynamic and then later there's some kind of structure that emerges in the consolidation? Like
You're thinking in the sense of like
where
if you
is STDP? I know that triplet STDP is this computational model that involves whether you get two spikes on the presynaptic and one on the post, which causes excitation, and two on the post and one on the pre, which causes long-term depression. And so I understand that as a kind of a model, but how would that model be physically instantiated? Does anyone have a sense of that?
I mean, Randall's the engram, the emulation expert here. But I guess in a broad sense, the way I would think about it is there's like homeostatic intrinsic mechanisms for the neurons that you're wanting to model. And then there's also the like different specific weights that connect the different neurons together that you want to understand, like that change over the course of plasticity and learning. So you need to have some understanding of like, well, how does this particular neuron connect to that neuron? How have its weights changed over time? But you also need to understand what are its like intrinsic electrophysiological properties. So like how long is its refractory period, or how fast is its like what's its basal firing rate, even in the absence of inputs, those sorts of things. And there's just like a whole heap of like different properties that you would need to characterize and understand. And I'm not really clear to what degree those differ between different neurons and to what degree the differences between different neurons can be read out of morphology as opposed to like electrophysiological characterization of that population type and other sorts of experiments. That's like a very high level overview. Randall,
No,
can maybe give you a more
that's very
specific
helpful
overview?
to me because I was groping to understand, like you're saying, the dynamic properties that lead to the structural, the consolidation, the structural consolidation, and I guess that paragraph that transmitter-reduced plasticity prevent neurons from becoming silent. That was a very interesting sentence to me because it seemed to indicate that transmitters are actually part of that whole dynamic process too, and will end up being abstracted away in pulling memories out. I don't know if I can. I don't know if anyone can. I don't know, in Randall, if you kind of have that intuition that transmitter.
I didn't read it carefully enough. Sorry, I would have to read it very carefully and look at their method section to figure it out properly because the way I'm seeing it right now in that sentence, I can't tell if this transmitter-induced plasticity is referring to anything different than the transmitters of the synapses that are experiencing triplet STDP, which I'm also not very familiar with. It sounds to me
what it's referring to is that
rather than timing, but
it's referring to the non-Hebbian terms in long-term synaptic plasticity, like two sentences earlier. So it's referring to the excitatory synaptic plasticity mechanisms, and saying that there is this whole set of complex ones. And I'd never understood that neurotransmitters were actually potentially just part of that system, a molecular signaling system that's just part of that active dynamic system that may be not so important for the for the memory itself. Anyway, I'm sorry.
Well,
Yep.
I think this is a good question in general, and there's a mechanisms review article that I was thinking about suggesting for a journal club that includes spike time dependent plasticity and actually behavioral timescale plasticity, which is actually very relevant to this in the hippocampus, and mechanisms behind those. So I think it might deserve its own journal club.
Thank you.
Cool.
Thanks, Ariel. That was great.
No, thanks for coming along, both to all the old faces and some of the new names. So I think Ken mentioned, so yeah, I might be presenting the next paper in a month or so in April, but we might switch over to Ken because I'll be in Japan at that time point. But I'll definitely come. I just might not be presenting. But if any of you are keen to present a paper at some point, then please feel free to message me and sign yourself up. Otherwise, we will try and keep these coming. We'll also, as time goes on, try to get more specific into papers that we think are good candidates for one of the annual research awards. So, for example, this paper I think is like very interesting and very fascinating, but it probably doesn't rise to the level of like previous winners for this particular award, because I'm not sure I like really have a much clearer idea of how to read out memories from a static brain after having looked at this, even though it is relevant. So if anyone wants to also nominate any particular papers for the awards or that we should be discussing, please feel free to do that too. Cool. Well, thanks for coming, everyone, and we'll see you next time. Bye.
Thank you.
Thanks, Ariel.